Good morning, everybody, and welcome to the Wednesday, October 23rd session of HNCS 7890. Today, I am pleased to introduce our speaker, Dr. Adam Cobb. Dr. Cobb's passion for soils emerged 17 years ago while volunteering on farms in New Zealand. After his time in grad school, he taught numerous courses at Oklahoma State University and contributed to more than 20 research publications. In the past few years, he focused his attention on public outreach and science communication. His new project, the Holobiont Hub, is set to launch in late 2024. Dr. Cobb, take it away.
All right, thank you so much. And it's great to be with you all. This is my, the ninth day since I came back from my trip to Spain. And so if we notice any typos in my slides, please let me know so I can fix them, but I'll just blame sleep deprivation. I'm really excited to speak with you today about holobionts and why I've come to believe, why my research journey, my academic journey, and personal life journey has led me to being so celebratory of the holobionts as really one of the key foundational elements to life on Earth, to our entire biosphere. You can reach out to me at these places I've listed here, and I'll see about getting the slide deck over to you all in case you'd like to check anything out. As mentioned in the bio, really my soil love story sprouted in New Zealand. At the end of university, I moved to Chicago and worked at a nonprofit for a couple of years, and then I was very, very burnt out from that experience. And I decided to do, what's the opposite thing I could do? I went to New Zealand and volunteered on farms and had lots of great experiences. You might note in the picture that I actually still had hair back then, a little bit anyway. But I really got the chance to experience these diverse land systems and sort of the lifestyle around, many times small scale, up to medium scale, but the agricultural kind of essence of what families were trying to do. And through my conversations with producers there in New Zealand, I really became convinced that if I wanted to reorient my career, which I was very interested in, that soils would be a great choice, and it indeed has been. So this is really just to explain, this has been quite a global adventure for me. I finished my master's degree in 2011. It was a non-research degree in agriculture, and that was really my way to get onto the track of sort of learning a diverse array of topics, maybe taking a course in post-harvest physiology, and then one in agricultural ethics. And then I had a course with Dr. Gail Wilson in restoration ecology. And I enjoyed that so much that I made the weird decision in my late 20s to change my path and become a scientist. I joined her lab in 2012, and then about 4.5 years later, I successfully defended my dissertation. So for those in the audience who are in that process, I was grinding my teeth at night, waking up with a sore jaw to get that dissertation done. But May the 4th will always ring out in my mind as the day I finally had that piece of paper saying I was a doctor. I rolled right into a postdoctoral fellowship, received funding through USDA NIFA, for that, and taught several courses, especially in food security and environmental science there at Oklahoma State. Continued to do research. There's a picture of my advisor and a good friend from my lab and I in Botswana, where we did some research in 2018. And I'm smiling in that picture because I'm working with cassava for the first time down in Central America in that photo. So soils can be a really great opportunity to take you around the world. My perspectives have shifted again in recent years because I moved to Oregon and I actually found my way to Dr. Elaine Ingham's Soil Food Web School. I was there for 3 years, met a lot of wonderful people. I've since moved to Idaho. That's a picture of my pup and I here in Idaho. And these experiences being back in the northwest part of the country and interacting with some very different folks than maybe I came across typically in Oklahoma, really helped me to expand my vision for what plant microbial communities look like, the dynamics, what we know about them, what we don't know, which I would say might be 90-something% of the story of soil that humans simply don't know yet. And that has culminated in me launching this, well, I'm calling it a co-creation community. I want to put things in the Creative Commons and get information about holobionts and soil biology out there into the world. So I want to give you a brief outline of my talk, the rest of my talk today, because that was all sort of intro that I had been asked to develop. I had a series of experiments with mycorrhizal fungi, and that was the crux of my dissertation. So I'll sort of show that 10 years of my academic research career, at least some of the highlights. The main one being the title of my dissertation was From Soil Ecology to Human Nutrition. And so I'll make that connection and we'll go into that data a little bit. And then I want to mention a few other bricks in the wall, things that built towards my perspective there through that season of my life. Ultimately, there's a paper that we produced a few years ago where we made a lot of recommendations about the ways crops would be bred. And we worked with a breeder. It was not just us naming and shaming. Crop breeders were big fans. So I want to highlight that as sort of maybe the more important, what I would consider the most important contribution I've made to the scientific literature. And then I want to launch from there into why that led me into this framework for holobionts, define the term, which you may have heard before, and then really talk about where I want to see us, humans, go from here, how we could get to a place where our current land management practices, which have many unintended consequences, could be improved by managing holobionts. And then I want to talk a little bit about regenerative techniques and approaches. This has been the world that I've existed in the last 3 years, especially. And to me, regeneration is not a dirty word, but sometimes it's a very flawed concept. And the way it's presented, especially back to academia, misses some key spots. So I want to talk about those. And then just finish by saying like how I'm looking to the future now. So the first thing I want to do is make sure that we sort out a term that I see used problematically all the time. So when I talk about mycorrhizal fungi, mycorrhizal relationships, mycorrhizal refers to the idea that there's fungus, and rhizal, root, fungus and root relationships. There are many different kinds, and they span different sections of the fungal kingdom, basidiomycetes, ascomycetes, etc. And they span different host plants. The orchids, for example, and the ericoids have a very unique form of mycorrhizal relationship. So a lot of times when I mention fungi, people automatically go to mushrooms, fruiting bodies. And one of the most charismatic is the Amanita muscaria, or fly agaric, which makes that beautiful kind of Santa Claus looking, possibly the origin of the Santa Claus color scheme. And that is an ectomycorrhizal relationship. It is a little less intense, might be the right word. The roots of a birch tree or a pine or several other species, I believe, can serve as hosts to Amanita. They will trade some sugars excreted from the root system to these fungi in order to gain benefits, usually improved uptake of nitrogen in those forest systems. That's very common relationship around the world, but ectomycorrhizae probably represent 2 to 4% of plant species on Earth. Even though they're charismatic, they make these beautiful mushrooms. The AM fungi, arbuscular mycorrhizal fungi that I studied, reproduce very differently. They form spores underground through their hyphal network. They don't make fruiting bodies. And so in some ways, they're a little bit more difficult to assess. We can stain the roots, as I have a picture here with a blue dye, and assess the area of the root that is covered with AM fungi using a microscope. This represents maybe 80 to 90% of plant species form that relationship, at least at some point during their life. There are some plants, such as oaks, that we think may form both types of relationships given circumstances. So I'm making sure to disambiguate, is my complicated term there, between types of mycorrhizal fungi, because a lot of times people assume that, for example, in a compost pile, you could increase the population of AM fungi. You would have to be growing host plants to do so. They don't just spontaneously reproduce, or they don't have like saprophytic capacity to really get food outside of the host plant. This is one of the papers I would tell almost anybody who's interested in AM fungi to check out, and a really nice diagram that shows a concept I'm sure you're somewhat familiar with, which is that we get this depletion zone around the roots of plants. Minerals, nutrients, I should say, that are relatively mobile in soil. Many of the nitrogen compounds, for example, may not be depleted. However, relatively immobile nutrients, such as phosphate, once the plant has taken up what it can easily capture near its root system, it becomes, the concentration dips more and more, and it becomes harder and harder for the plant to find adequate phosphate in this case. So you can see these pink structures. They'll actually connect to the roots of plants. They make a site of nutrient exchange between the plant and the fungi. And they can extend the reach of that root system so that there's a larger volume of soil and that there's a finer scale of access. So not only with phosphate, although that's one of the largest, but with zinc and we believe calcium and many other limiting nutrients, the fungal network, the hyphae, really gives the plant an additional edge. It can give plants competitive edge over neighbors that are not as reliant on AM fungi. And then indirect benefits, almost any fungal entity in the soil will improve aggregation of the soil, will help gather those small microaggregate crumbs of soil together and weave their thread-like structures around them to create a more stable aggregate, which can open up space for water and oxygen or air to move through the soil. You can also get more carbon accumulation in systems that have a lot of active AM fungi because the cell walls of the fungi themselves are somewhat resistant to decomposition, so they can become a sort of a sink for carbon. And also, because of aggregation, you can get sort of the entombment of particulate organic matter, at least. And so that those can go for, they can be in the soil bank for dozens, if not hundreds of years. And then all in all, you end up with a lot more stability in these kinds of systems. A lot of our inspiration, and especially coming through Dr. Wilson's lab, was to look at highly functional tallgrass prairie ecosystems and observe these relationships there. And then I was the weirdo in the lab that said, can we apply this in food production settings, in not just in ranching, because that's obviously an important place to manage grasslands, but also in row crops and in other aspects. Okay, so the question was pretty simple. Can we unleash this highly mutualistic relationship? And I include this picture. I don't know if you'll see my pointer, but it does a great job of showing how those spores are formed off of that fungal network. And these are really interesting biologically. It's quite clonal. There is recombination of DNA, but we don't really understand how that's happening. There's the fusion of different individual strands of hyphae so that offspring have a mixture of genetics, but it's not like typical reproduction. It's fascinating to look into. But on a practical scale, I've included this picture of a alfalfa study that we did because this was a classic thing that was done in our lab in many mycorrhizal labs. We steamed the soil here so that we would get a kill rate that was very high of all the organisms in there. We put back a sort of a microbial wash where we didn't allow any of the AM fungi to pass through the sieve. It was too small for them. And so our hope is that we have a lot of the bacteria and other things here in the soil system. It represents, relatively what these plants would encounter in the field, minus AM fungi. And these guys in the front, alfalfas that don't have their AM fungal partners, as compared to the growth of the same variety of plant in the background, the same conditions, you can see that there's a huge growth response to this partnership. I didn't use alfalfa primarily in my research. I used a lot of sorghum bicolor. This is actually me in southern Oklahoma harvesting one of my plots. And this is just because sorghum is not only an important grain crop, it's produced a lot in the world, especially in areas with limited resource use, but it's also related to a lot of other crops and grasses. We can get information from genome studies and get an idea of the functional genes that are maybe related to, for example, mycorrhizal symbiosis. By examining sorghum, it's a lot less complicated than some of our crops, and yet it's information gained from sorghum can still be insightful to corn and wheat and other grain crops especially. So here we're finally getting to the first data set that I'm going to show you. This is a paper that we published in 2016. It was my first paper. It was the first chapter of my dissertation. And I don't know if we have too many professors in the room with us today, but you're going to laugh at how much I was spoiled. I think I sent this manuscript away in July, and then I went to hike in Peru with some friends and celebrate graduating. And I came back and I had the paper published by October. So my peer reviewers were on it. They did 2 rounds of reviews really rapidly. We were able to make the changes. And then the next paper, I had to wait 6 months for my initial review. So I got a little spoiled here. But this is going to show you, where we started to really dig into this question of how the genetics of the plant could influence the relationship with AM fungi, and in turn go all the way back to influencing the grain quality and quantity. And so I want to orient you to my first graphics here, because this is a concept, it'll come up one other time later in the talk, of what we call a mycorrhizal response. And you mentioned if you have that little plant and that big plant, you kind of figure out, okay, if we think the major factor difference here was the AM fungi, how much are they contributing to this growth? So what I have here in green are, we called them inbred genotypes, but you could also think of those more like a landrace variety of sorghum. Some of them were developed in sub-Saharan Africa. Some, I believe we had at least one that was developed in India. And then in blue, we have these hybrid types, what you might buy at the store if you're putting sorghum in the ground as a US farmer. And hybrids have a lot of value as a technology, drought tolerance, disease tolerance, there's all kinds of things. But we had been speaking with some researchers in Ethiopia at a conference who said, hey, when we get these commercial sorghums from the US, we can't even make them grow on our research station to their full potential. We don't have the fertilizers and other resources. And so we're not going to give them to our small scale farmers because they simply won't produce. And we started to say, what if there's been an inadvertent loss of the capacity of the plant to host AM fungi. So we grew these kind of pairwise comparisons of the varieties, and we also had a fertilized and not fertilized system in the research. And we also used fungicide in one case to completely knock out the AM fungi from that system as well. But the first arrow I want to mention that's here on the left-hand side is showing you that in terms of grain production and overall growth of the plant, those hybrid varieties, when they aren't fertilized, they do indeed produce very poorly. And those inbred genotypes or more landrace style sorghums were still quite productive for us. And then if you jump over with me to the right-hand arrow, we found that relationship might be explained below ground. The inbred genotypes were still hosting 60% of their roots were covered with AM fungi, and maybe only 20% or a third of that in those hybrid genotypes. So this got us thinking about what genetically may have been lost here, right? Now, when we fertilized everybody, they all did about the same, and they all greatly reduced their mycorrhizal colonization. That's typical, especially high phosphate concentrations. The plant will downregulate its relationship with AM fungi. But what kind of made my project a little bit more unique, this kind of work has happened with different crops before, especially wheat. My advisor had done some papers in the 90s. But we wanted to go all the way to grain quality, and we were working with a USDA lab that specialized in that. So I ended up saying, hey, indeed, the protein, phosphate, or phosphorus, zinc, calcium, all of these minerals, they are highly correlated to the relationship of AM fungi and plants that we can observe in that root system. So we use that root colonization to sort of see where these results fell. And it did indeed seem that if you're going to hand a variety to a small scale producer, you would want to hand a variety that you knew had a high propensity for partnership with AM fungi, especially if that small scale producer cannot access all of the irrigation and nutrient supply that we might be accustomed to in our breeding programs. So this led us to this conclusion in the paper. Again, I'm not putting crop breeders on blast here. I'm saying maybe we have accidentally selected against hosts, plant hosts that can signal to AM fungi, maybe even that they can recognize their own deficit in some of these resources because we have bred them generation after generation in quite luxurious conditions, lots of water, lots of nutrients to control for those factors while we're on the pathway towards disease resistance or drought tolerance or something else. And so it's just a unintended consequence is kind of what we said in that paper. So I'm going to ask you to hang in with me here. We're making good time based on what I have, but I want to share a few other highlights, especially for those of you who may be looking at a research career. You might find it interesting the pathway that I went on here with publications, because I mentioned this metaphor of building, like putting bricks in a wall, and each publication really set the stage for the next question, the next project, the next grant. And that's kind of how it goes. This one, I published only because Gail, when I was headed to visit a friend in Zambia who was in the Peace Corps, said, oh, I have this international soil import license from APHIS, and I want you to bring back some samples of some of the different smallholder farmer practices or some of the fields that were treated differently or managed differently, and we'll see if we can do a small write-up on that. And we did indeed get this published. This was rural eastern Zambia, almost Malawi, if you went a little further. And I was able to use phospholipid fatty acids and neutral lipid fatty acids, which are not a perfect measurement tool by any means for microbial biomass. The biomarkers do tend to work pretty well for AM fungi. We can isolate that, a particular biomarker, and do sort of a transformation of the concentration that we get so that we can eventually get to a kind of a biomass measurement, abundance, right? And I'm really glad that I threw in this local grassland control because that ended up being a lot of the story here, regardless of the methods, and there were different farming methods. There were very low input farms. There were farms utilizing that had been trained in agroforestry. There were farms doing very high input of subsidized fertilizers, and there was increasingly conservation farming going on, especially reduction in tillage in the area. And all of them had a marked decline, maybe half of the fungal biomass. Conservation farming seemed to be allowing for the recovery of a spore bank of AM fungi, but we still weren't seeing a ton of active AM fungi. And this fits with other research around the world that tillage and fertilization and many of the other common agricultural practices in the world can be very disruptive to the AM fungal community. And you end up with a decline in diversity, decline in abundance in soils worldwide. These studies have been done in Europe and the USA. Probably the coolest thing about the Zambia study is just that it was done in Zambia, and there aren't that many research papers coming out of Zambia these days. I want to mention just briefly another paper that I was invited to. My friend Shashir was actually a postdoctoral researcher across the hall from me, and he had this project with a bunch of USDA experts on grassland management and comparing outcomes. And it ended up being a huge part of the framework that I developed, while writing this paper with Shashir and going through revisions. It took us a year and a half or something from start to finish to get it published. And I'd say one of the big takeaway messages here is we found this idea that there is quite a continuum of grassland management styles. And I used to think much more black and white, like you have your chemical tools on one side, you have your biological tools on the other, and they don't really blend. And I found out that actually in real agricultural settings, people are blending, they are attempting to build up ecological management resiliency, but still reaching for those chemicals as needed. And so that's one of the tricks in the transition there is to find that sweet spot of intermediate management that's really, I'm not such a hippie that I'm against ever using something from the agrochemical world. I do think that there can be big unintended consequences to water quality, to soil health, and we should understand that and use them very judiciously. I will mention as well that I got very lucky with something that happened in 2016-17. There was a large research group at the China Agriculture University where they had sent one of their star pupils our direction. She stayed with us for a year. We struck up a friendship, worked together on research, and eventually published that alfalfa paper that I had the picture to. But we were also invited over, Gail and I, twice to China, to Sichuan Agriculture University and to China Agriculture University in Beijing. And something like 15 or 16 papers came my way out of that expanding collaboration, published just earlier this year with that now professor who was the PhD student that came to work with us. That's me in actual Oklahoma State swag, standing at the Summer Palace outside of Beijing in 2018. And yeah, this is, I would just encourage anybody who is thinking of a career in science and research, that collaboration is going to be really your bread and butter. There's going to be so many opportunities that come to you if you are a good collaborator, and I'm really grateful for what I learned, the experiences I had, and the way that it bolstered my CV. So I want to do one final paper here before kind of heading towards the summary of this talk. This is the one I mentioned might be, I might consider it my greatest contribution to the literature. So I spoke of mycorrhizal growth response before. You can also do a phosphorus response by looking at the total phosphorus in the plant tissue. And we now knew that crop breeding could take us the wrong direction. There's other articles and books out there that mention that. But we wanted to work with this breeder, Dr. Wu, that was there at Oklahoma State. He had a big project doing switchgrass breeding. Say, can we go in the other direction? Can we increase the reliance of the plant on mycorrhizal fungi? And so we wanted to track not only the growth response, but also the phosphorus response. And there is an equation in the literature that we can use. I mentioned this before, so we end up a lot of times with these little plants. These are some of my sorghum, and these bigger plants. And we want to do a comparison so that we end up saying, in this case, you end up saying, how much more did the plant grow because it had its fungal partner? In ecological studies, sometimes you say, what percentage, like so it's scaled up to a max of 100, what percentage of the growth of that plant came from fungi? Slightly different. But this is what I'll use here. I just have a couple of data sets to show you. So when you calculate mycorrhizal growth response this way, you can get percentages in the thousands, right? Because if you have a tiny plant and a big plant, the big plant is thousands of times bigger than the tiny plant. And what we found is that these lines that Dr. Wu had been breeding, these switchgrass lines, compared to their parent lines, were performing better in these conditions, right? So when we didn't give them tons of fertilizer, when we assessed how much they kind of reached out for help from fungi and found that relationship worked effectively, then we were very impressed with how much larger they grew as compared to their parent lines. And we found something very similar with that mycorrhizal phosphorus response, that the total phosphorus that had been pulled out of the same soils, the same conditions, was greater in these lines. Now, that brings up a whole different range of questions about management of those systems, but those are kind of agronomy related more than anything. What's the right rate to put in to not disrupt this relationship, but to keep the whole system working optimally? These are big questions. I will point out one other thing. Sometimes peer reviewers drive you crazy. I've received peer reviews from grant applications and from papers that made me very upset, that seemed like somebody was very closed-minded or had judged ineffectively. But sometimes reviewers really help. And something Gail has mentored me a lot in over the years is try to look for the way that they're helping you improve your work so that the final product will get more accolades from your peers, will be cited more often. And just about a month ago, I finally got around to listening to the entirety of Adam Grant's book on tape, or on audio, I should say. Think Again. Embrace the joy of being wrong, right? So what happened here is I had made a kind of a crappy graphic, and the peer reviewer didn't like it, and they said, do a deep dive on holobionts and see if there's something there in the literature that you can use. And I ended up coming up with this graphic that explains, on the left, sort of the traditional way of thinking about a breeding program, which is to focus on the plant's genome instead of the hologenome, or the entirety of all the microorganisms, including fungi. And that may be why we're kind of like tuning the selective pressure to something that has unintended consequences below ground, because we're not looking holistically enough, right? Holobiont, I'll get into this, comes from or is related to the term holistic. So in terms of microbial ecology, soil amendment use, and agroecosystem management, we put out there this idea that yes, there are many ways in which we are breeding crops for a particular context that's not necessarily the context we want to be in forever, and it's not necessarily going to be appropriate everywhere. And that was sort of the take-home message, right? So where did I go from here? This is one more picture I took, right, of big plants, little plants. So the same sorghums, I grew 50 different varieties in this research project with AM fungi in the soil ecosystem, and then where the AM fungi had been removed, and you can see that marked difference there. Okay, so I got really interested in agroecology at this point, and it's like sort of broadening yourself if you have focused on being a soil microbial ecologist, to think about these messy human systems. But it works. Going all the way back to that restoration ecology course I took with Gail, we ended up talking about economics and politics and stuff in that course a lot, because when the rubber meets the road, it's not just the science that directs human activity, it's also human culture, right? So I'm going to get a little further out there in terms of some discussion. Where did I go after my decade in academia? A couple places that I went. I ended up here in Idaho, and I'm using my own backyard as a big experiment for some of the things that I've learned from Elaine and others at the school. Uh, I will say there's probably a dozen things I could think of off the top of my head that I disagree with, that are sometimes talked about in regenerative agriculture circles. I'm quite willing to be the happy warrior over there because they're my people. I really believe that there's something that there's there. I just also think we have some evidence issues in a few cases. So the soil food web is where I've been spending a lot of my attention, and all learning about all these different organisms that often get reduced, like the ant fungi. They get less diverse and less abundant in the way that we're managing in modern agricultural systems. So the thing I've done is I've stripped that back and gone to the smaller scale in my backyard to prove some things to myself. Okay, I'll get into those details a little bit. I also, thankfully, have continued to be able to interact with producers at the larger scale. And I always wanted to be the kind of scientist who didn't just have a pretty theory that got me, some notoriety, but actually understand how ranching and farming is done. So I have interacted with these groups everywhere I've lived. This is actually from about a year ago. I made my first compost pile the way that Elaine Ingham would teach it. And I did find some really interesting results that I'll mention. But to me, it goes all the way back to this, what Aldo Leopold called the land ethic. I believe no one else, this was, in the 30s. Highly recommend if anybody wants to check out his book, The Santa County Almanac. He was a scholar, a poet, a farmer, an ecosystem thinker. And he realized that our relationship to land and soil had been kind of corrupted by an exploitation mindset. And that's really where I've ended up as well. It's not just that I think that there is some scientific validity to managing ecosystems and holobionts the way that I've been talking about, but also that I think it's going to be better for us as a species. That here we are, maybe the last 100 years represents our adolescence, our growing up time as a species. And if we ever want to get to a more stable existence for ourselves, for our children, There's a lot of maturing that we need to do on the cultural level. Maybe not everybody. I can point to a lot of indigenous communities around the world that are interacting with their natural resources in a very positive way, but a lot of us with how we live. So what's a holobiont? So I mentioned that word holistic, holobiont. I sometimes switch back and forth, by the way. Hollow. um, I love ruminant animals. These are pictures that I took on farms or on ranches in Oregon. Uh, they're a great example to me because if you kill off their microbiome, their gut organisms, they cannot get the nutrition that they need from what they eat. So you have this host, it's the goat or the cow, that creates this container, and then there's all these other organisms living inside of it and on it. I read a paper just recently that says we don't realize how much we as humans have microorganisms all over our skin all the time, inside, nematodes and other things crawling around, right? So we are also a holobiont. Um, and it's if you look at the host organism and try to manage that or breed that in isolation or ensure that it is healthy, but you don't pay attention to the other organisms there, I think you're really setting yourself up for failure and for sort of this treadmill of micromanaging and dealing with problems and doing things that have big unintended consequences because you have to, right? So we have tuned our land management, food production systems in many cases towards plants and animals instead of towards holobionts, instead of towards plants and animals that have these vital relationships with other organisms. I want to make this practical though. So you may have heard, and this has been used in pandemics and other times by the epidemiology community, this concept of the disease triangle or disease. I know this is not a triangle, kind of a triangle, the way I've made it. You can imagine one there. But this theory says you really have to have favorable conditions for disease. You have to have the presence of a pathogen, and you have to have a host that is susceptible for the individual to become ill. So disease happens there, and it's pretty small overlap of all of those conditions. If you have a host that's no longer susceptible because you immunize them, for example, then the pathogen can be all over the place and conditions can be favorable to a person becoming ill, and the host may resist it still. Disease wouldn't happen in that case. So I want to use something similar to talk about this holistic theory. I believe that if we make sure that we have all of our mutualists, which again, many of our common agricultural practices reduce or eliminate mutualists. You will not find that many protozoa and beneficial nematodes and fungi in soil that's been tilled and fertilized and sprayed with herbicides and treated this way. So you need the mutualists. You need an effective host, which could come in to the breeding question. Are we breeding effective plant hosts? And then you need a favorable environment. This can be things like enough moisture, the kind of Goldilocks zone of pH in the soil, the presence of air exchange, so that you have, if you have mutualists that are, for example, obligate aerobes, such like many of our plants, that they would be able to find sufficient oxygen to keep their systems running, right? So these thriving holobiont communities, it is a community, also have a relatively small overlap in these conditions. That is what I'm trying to manage for in my garden. That's what I'm trying to find examples in larger agricultural contexts where people are setting the stage for thriving holobionts, because all kinds of things that would hurt our desired plant species, our animals, can be overcome, fought off, by the highly tuned immune systems of effective holobionts, right? By the favorable, the environment tilts the stage away from disease and towards health. So what does this look like for me? Again, I mentioned that there's this whole soil food web. I'm holding here some of the compost that I made, and then I decided to feed to composting worms for a few more months. And when I slap that on the microscope, I see approximately 17 gajillion protozoa and nematodes. And sometimes I spot a tardigrade in there. And all of this evidence, lots of fungi, all of this evidence of a thriving, teeming, diverse soil ecosystem. If I were to go and get soil from the farm down the road that is managed the way that people have been managing corn crops in monoculture for a long time, I think I'd find a few bacteria, maybe not even bacteria that I could surmise were beneficial to the plant. I don't think I would see a lot of fungi. I don't think I'd see a lot of beneficial nematodes, protozoa, other microfauna, microarthropods, earthworms. These things are harmed pretty directly by the ways that we manage soil through the chemical paradigm. So I'm coming close to the end here. I know I was told to do about 45, 50 minutes. This is one of my favorite quotes. We often forget that we are nature. Nature is not something separate from us. I see a lot of worship of this idea of the natural in my field. So when we've lost our connection to nature, we've lost our connection to ourselves, right? And it again goes into to make the statement, oh, we've lost the protozoa and the aim fungi and whatever from this field, but they're not important. We can still produce food there. We just have this high input system and high output system and all. I really think that that misses the mark about the way that humans and the rest of the world can be a kind of a large scale holobiont. We can be an interdependent biosphere. So picture's worth 1,000 words. I pulled this off of a water conservation district post on social media where they compared soil from fields that had been treated in a conventional way with chemicals and tilled and all of that. And they really had unstable dirt, I would call it. When they put it in water and did the slack or slake test, it just fell apart. And then they had an example of a farm that had switched to many of these ecological and biological methods, reduced their tillage and made sure that they weren't putting excessive salts into the soil through inputs, and they had resilient soil. They had soil that could hold up to being inundated with water. I'm doing this again on small scale. There's Riley. She is my little helper here with the backyard garden. I think that everybody should have a backyard garden if they have enough space, or houseplant, or whatever you can do to fill your life with relationships, not just to plants, but to the holobionts. I'm taking worm compost and treating my plants with it and finding that they are very healthy, resilient, resistant to disease and insect attacks. I'm trialing these things on a small scale, but I'm also seeing ecological management at large scale. One of my favorite stories is Adam York. He's a producer of corn and soy primarily in southern Illinois on thousands of acres, tens of thousands of acres, rented land and owned. And he had large pivot irrigators and was doing the whole regime that is quite typical in those systems. Got into the soil biology, compost biology side of things, retooled his pivot irrigators so that they could deliver that that biology from the compost as an extract to his crops, has seen his input costs reduce $2 million in the first year, has, if anything, seen yield increase over time, water holding capacity in the soil. This can be done at scale. So I'll just end by mentioning We often summarize these regenerative principles. You can read about them online. I like this because it gives us the chance to say there's the principle and then there's how you apply it. And it's going to look different in a lot of places. One of the things that I studied in grasslands, plant diversity almost always pays off in terms of ecosystem services, in terms of yield and quality, in terms of animal gains. And so I very much believe in the goals that are put out there. I can again talk about some evidence that's lacking, and I'm aware of that. I wanted to mention I also just got back from hiking or walking the Camino de Santiago in Spain. This is me and several of my friends ending our journey in the rain there in the final city. I'm still searching. I'm looking personally and professionally for these kinds of answers. The transformation of human society is a pretty big goal. And I don't think I'm going to do it alone. But I believe that in celebrating holobionts and in building systems around them, we can come a lot closer to where we need to be in terms of our human culture as well. And with that, I will take any questions.
All right. Thank you, Dr. Cobb.
Cousin Adam.
Pull up a question. Actually, if anybody online wants to unmute yourself and ask the question directly, feel free.
I have a question, if you don't mind, Dr. Gardner.
Go right ahead.
Thank you very much, Dr. Cobb. Appreciate your presentation. I guess I was just wondering, in your framework, how do you recommend replacing nutrients that are exported just kind of by default from a farm that exports crops? Like, what would your recommendation be?
That's great. There is a wonderful paper, I believe one of your researchers there at Ohio State was involved, all about that ecological nutrient management system. So yes, we are. Let's talk about scale. If you are exporting beef from your property, you're not exporting tons of nutrients in the animals. If you're exporting hay, you're exporting a lot of nutrients, right? As it's drawing them from the soil every year.
Right.
So there's a scale difference there. We need to understand the nutrient inflows and nutrient outflows and track that very effectively. When you switch over to a mentality to put the nutrients in mostly in the form of organisms, like bacteria and fungi that are living off of organic matter in a compost pile and then going into the field, it's really an efficiency question. So I've done a lot of study of the way that we typically fertilize, sort of the green revolution style, and it's like over half of what we're applying is typically wasted, right?
Mm.
It goes into the air as nitrous oxide, a greenhouse gas. It goes into the water supply and causes eutrophication. So again, I'm all about those intermediate systems, but I think that some blend is necessary there. And I am critical a lot of this thing where organic certification was like, you can't use any of these chemical fertilizers or commercial fertilizers, because I'm like, okay, if you're just adding animal waste, then maybe that's like 4% N and 4% phosphorus, right?
Mhm.
In chicken waste. But the phosphorus is not going away as quickly. So you're accumulating like a toxic level of phosphates in your field
Mm.
in order to meet the nitrogen needs of your plant. Like, I would love to see people be okay with supplementing with some urea or something, right? If they need to. But I've also seen that you can transition these systems over a few years to where they're just not addicts to the fertilizers, right? There's an amazing capacity if we bring in legumes and other techniques to keep that nutrient balance there. So I don't have those answers in all cases. I just, again, would pivot to that principle and see if I can find that paper on ecological nutrient management for you.
Yeah, sure. Sounds good. Thank you.
Thank you.
Do we have questions from the live audience in Columbus?
I just want to say hi to my cousin before he signs off.
Hey, Amy.
Hey, thanks for speaking.
Right, that
That was
works.
awesome.
Some questions from online. What are the avenues for promoting a land ethic and considerations of holistic management in places like the Midwest where 85% of the land is in row crop agriculture?
Yeah, that's a good one. In general, because monoculture is like one of the really tricky things to get around, it is a big challenge in those areas. You could always claim that this is quite biased of me because I was a prairie ecology enthusiast too, but I might say a lot more of the Midwest should be in perennial, diversified grass ecosystems and managed that way. And row crops can be quite destructive if the slope is too extreme. It can cause a huge amount of erosion. We see that sometimes through loopholes in the subsidies that end up happening, that people plant more corn, fence line to fence line, we call it, than they know will actually produce so that they can claim those subsidies, right? So, but let me get directly to your question. I have been very encouraged to see a lot of young people, I'm saying 30s, 40s, without historical access to land, especially in urban and semi-urban areas, uh, find ways to create farm to market businesses that are really effective. And one of my favorite examples of a producer that got this right is a gal in California with such a business that was like she was doing all the chemical pathway. Every solution has a chemical to buy, very expensive. And she ended up working with a guy that I know who's a consultant. And he taught her, he's like, you have these below ground livestock. If you're producing tomatoes or whatever, there's microorganisms there. They're really, really tiny goats and sheep and cattle, right, that live in your soil. You need to work on them, on making sure they're healthy and thriving. And then they will produce really great tomatoes that you can sell at market. So I do think that a lot of our row crop agriculture needs to be transformed into more highly diversified, high nutrient supply. I think we have been kind of addicted to this commodities thing for a long time. And, they say that like medical teachers tend to teach students to solve the problem that is no longer a problem in medicine. And I think the same thing happens in agricultural settings. We were really worried about having enough calories for a long time in human history. And we have enough calories now. We don't have adequate nutrition. We have a lot of double burden of obese populations that are also malnourished. And so we've got to look at these systems and say, should they be grassland systems with animals on them? Should they be diversified farm to market vegetables instead of row crops? And I don't know how all that happens without creating a lot of chaos and pain in human society because we're kind of tuned to having these cheap commodities. However, it's going to happen no matter what because of climate change. Like, farming will not be able to be done the same way, in my opinion, in 20 years as it is today. I hope I answered your question somewhere in there.
Very nice. In the interest of time, Dr. Cobb, do you mind if I provide our online users with your email address in an announcement, and then they can ask you questions about your seminar that way?
Sure thing. Yeah, and I can probably send some papers around to folks that way too.
All right, that would be fantastic. So those of you that I didn't get to your questions, just go ahead and copy paste those into a text file. I'll have Dr. Cobb's address to you in an announcement in about 15 minutes. Otherwise, Dr. Cobb, thank you very much for your presentation today. That was very interesting, and I'll see everybody on Friday.
Appreciate it. Cheers.